Sir, you can start.
Yeah.
Himanshu sir, please.
Yes, madam. Yeah. So very good afternoon, everybody.
Good afternoon.
On behalf of Hindi Vidya Prachar Samiti's Ramnirajan Junjunwala College, I extend a very warm welcome to everyone for this popular lecture series number 7 under the DBT Star College activity of our college in association with Association of Teachers in Biological Sciences. It's indeed our great pleasure to have with us today our guest speaker, Dr. Narayan Punekar, who is going to deliver a talk on enzyme catalysis. Body functioning requires energy, and that energy comes from metabolism. What lies at the center of the metabolism is enzymes. Well, when I have learned about enzymes, or when I teach about enzymes personally, uh, I find a very spiritual angle to it in a way that I think enzyme functioning and catalysis lives totally to that one saying in Bhagavad Gita, Karam kiye ja, phal ki ichha mat kar. It keeps on converting a substrate into a product, but while doing so, with its dynamism, it opens so many secrets to us. And Dr. Punekar has a wonderful method of explaining this difficult topic and understanding, make it easy for the topic with his very unique and lucid manner. We have had opportunity of listening to Sir at Junjunwala College at the earlier occasion, but today, like me, like Madam, like Kale Sir, many of you, which includes the members of ATBS, which includes the members of teaching staff, our PhD and postgraduate students, our well-wishers from Pune, Delhi, in front of all of us. We all of us together are going to enjoy this lecture. So thank you very much, Punekar sir, for accepting an invitation for today's lecture. And now I request Kale sir to give a formal introduction of Sir. Kale sir.
Namaskar. I'm Professor Purushottam Kale, and I'll introduce to you briefly today's speaker, Professor Narayan Punekar. Professor Punekar is a professor biotech group in the Department of Biosciences and Bioengineering, IIT Bombay. After his graduation and post-graduation in biochemistry from Dharwad, Karnataka University, Professor Punekar did his PhD in enzymology and microbial biochemistry from Indian Institute of Science, Bangalore. He was a postdoctoral fellow at Institute for Enzyme Research, University of Wisconsin, USA, from 1983 to 1986, after which he worked as a research fellow at a School of Pharmacy of the same university from 86 to 87. 87 to 88, he worked as senior research scientist at the Research Center of Hext India Limited, Mumbai, and joined IIT Bombay in 1988, where he is till date. Well, in 1996 to 98, he was a career development fellow of Rockefeller Foundation, University of Wisconsin, USA. Between 97 and 98, he also received a fellowship for a collaborative research project with Professor Hines of Department of Genetics, University of Melbourne, Australia. 2016 to 17, he was the first Dean of Academic and Students, IIT Dharwad. Professor Punekar's research interests are in microbial biochemistry and molecular enzymology, microbial metabolic regulation, understanding metabolism through biochemical and recombinant DNA techniques, fungal molecular genetics, and its applications to metabolic engineering. Professor Punekar has guided over 45 students for MSc, and 20 students for PhD. He has over 50 research publications and 4 patents to his credit. His book on enzymes, catalysis, kinetics, and mechanisms has been published by Springer Nature, Singapore in 2018. He has received Research Dissemination Award of IIT Bombay in 2018, Excellence in Teaching Award in 2002 and 2012, Professor S.P. Sukhatme Excellence Teaching Award in 2018, all by IIT Bombay. He has been a member of National Scientific Committee for the International Chemistry Olympiad, which was held in Mumbai in 2001, and also for International Biology Olympiad, held again in Mumbai in 2008, when of course I was the leader of Indian team. Professor Punekar is also a member of editorial board of Indian Journal of Biochemistry and Biophysics, Indian Journal of Biotechnology, Indian Journal of Experimental Biology, and so on. Today, we are going to listen to him speaking about enzyme catalysis. So over to you, Professor Punekar. Thank you.
So shall we begin?
Yes, please.
Yeah, okay. Okay, great. So a very good afternoon to all of you. I must say that they have been excellent speakers. I have heard many of them on YouTube already. And I guess this is going to be the second Punekar for you, right? At this part of the series. So, but obviously the topic will be slightly different. So a slight change in the title, it's put as enzyme catalysis, but I would probably focus more on history of enzyme catalysis and also try to bring you up to date on where we stand today with respect to this particular field. Yeah, and obviously we'll have an occasion to have some question and answer at the end of it discussion. So what I will try to do is to show you particularly the biology audience. You know, there are students of biology who are a little scared of math, who are a little scared of quantitation, more of description. So for such people, I think quite often enzymology comes as a little difficult topic. What I would like to do is to show you that it's not as bad as you think. In fact, it is very exciting. Okay, so there is nothing better than going through the history of a subject to understand the subject better. Yeah, so what I will do is take you through the history of how enzymology evolved.
Sorry to interrupt you, sir.
Yeah.
The presentation is not being seen.
Yeah, but I will put it on, okay?
Okay, okay.
1 minute.
Thank you.
Okay, sorry, I'll put it on. Sorry, I think I made it. Should be there now? Can you see it now?
Yes, we can see.
Yeah, okay. So what I will try to do is to show you that history is a great teacher, and we'll look at enzymes through the historical perspective as a part of this, probably 40 minutes and then maybe some discussion. Okay, so this is Christian Didouet, who said that any living being is a reflection of its enzyme arsenal, and we are and do what our enzymes permit us to do, right? I mean, that's the famous biologist for us who said this. Yeah, so enzymes, when you think of enzymes, you also look at what they do. Basically, they do chemical conversions. So obviously, Book of Life is written in the language of carbon chemistry. So most enzymes that we see have to deal with carbon compounds and interconversion of carbon compounds, which is the basis of life in itself. Okay, so chemistry of carbon and biology. Obviously, the enzymes form a major bridge between these two fields, chemistry and biology. And one of the reasons why I guess biologists might hesitate to look at enzymes a little more in detail is because, you know, they would have forgotten chemistry a little bit while ago. But I think that's not a big deal. You can always pick it up. at any time. But enzymes being the key players, okay, chemistry is a prerequisite in some sense. So this is a Harris cartoon who tried to play biotechnology. What is the 9-letter word for biotechnology? Somebody said chemistry, okay. But then what about biology? All living systems are chemical engines. So you will figure out that they are open systems. So there are a lot of chemistry that goes on inside. Whatever you take in, whatever you give out, a number of steps. So every step is enzyme catalyzed. So you need to understand that. And obviously enzymes are the place to start this work, this understanding, okay. So this is a little busy slide, but I just show you this because I thought I would bring you up to date with respect to what kind of work has been done over time. So this is a slide showing all the Nobel laureates right from 1902 all the way up to as recent as 2018. Okay, these are various people who contributed to the advancement of enzymology, and obviously people got Nobel Prize for either for chemistry or physiology and medicine, which itself tells you that, look, enzymology is truly an interdisciplinary field where you need to have a knowledge of a bit of chemistry, and also it plays in the field of biology, okay. So you can see Emil Fischer, the first Nobel laureate, stereochemistry, lock and key concept, 1902. I'm not going to read through the whole of it, but just to tell you that the most recent, which probably 2013 was computational enzymology, and who looked at transition states of enzyme reactions. And then the most recent is 2018, just about 2 years ago, year and a half ago, Francis Arnold on directed evolution of enzymes. So this is a field which has really got great recognition and stalwarts to contribute to this field. Okay, so Perry Fry was one of the faculty at Enzyme Institute, and he has to say this about enzymes. Any biologist who follows their research interest to the finest level of detail will eventually become an enzymologist. Because I think the final step is actually a reaction inside a cell or inside a body. Okay, so also let me say that a lot of chemists have contributed to enzymology, and one example I can quickly take is that of Arthur Kornberg, who did a lot of work on enzymes, and there's a lot of nice background if you are interested to read from his book, more like a biography, For the Love of Enzymes. So some of you might be interested. Okay, so now I go to the proper history of enzyme studies right from the beginning. So landmarks in enzyme studies, what are the kinds of work people have done? So this also is a busy slide, slightly color coded, particularly those with blue, I will spend a little bit of more time. But as you can see that the oldest study of enzymes that you can think of was in 1751. We did not even call them as enzymes in those days. As I said, history is a great teacher. Riemer in 1751 studied gastric digestion in birds. Very interesting story, actually. You know what he did? He took pieces of meat, tied a thread to it, and fed it to these birds of prey, like vultures and falcons, and let it go into the body, into the stomach, wait for some time and pull it out. So he could figure out that the meat could be tenderized and digested in the bird's stomach. And obviously the next step was you go to the actual gastric juice, which Spallanzani did in 1780, and figure out that, okay, the digestion occurs in these places. And obviously then you see that gastric juice is a source for enzymes. And I'm sure today we know both intestine, stomach, you have enzymes like pepsin, chymotrypsin, and a whole range of enzymes. Similarly, in the 1883, amylase was discovered by Payne and Persos. He showed that it is exactly like actually hydrolysis by acid. So catalysis as a historical perspective began with these kinds of studies. And the first time that the concept of catalysis was described was Berzelius, famous John Berzelius, who's a chemist, also a medical chemist in those days. Okay, he was also a pharmacist. So he got the concept of catalysis. Many of biologists may think that catalysis as a concept came from chemistry. No, catalysis as a concept came from biology, particularly studying biological systems. Okay, and eventually you got into chemical catalysis for chemists. But as you can see, the earliest examples of catalysis were not in chemistry, but in actually biology, biological catalysis. Okay, so then Kuhn termed these molecules as enzymes, and then Fisher did lock and key concept. Then you get Henry's Michaelis-Menten equation. So a whole range of people have contributed. And obviously you can see Pauling, you see Westheimer, you see Phillips, structure of lysozyme, Merrifield, ribonuclease synthesis. So the most recent and probably one of the landmark studies was in 1980s, 81, which is catalysis by RNA molecules, Altman and Seck, so ribozymes. So the concept got a change that not all enzymes need to be proteins. Some of the catalysts can also be non-proteinaceous. So ribonuclease A was, ribozyme was studied in those days. Okay, and it was shown by these two people. Okay, so historically, I think there's a great deal of history as we know of enzymes. We knew about enzymes even before we knew they were proteins. In fact, we knew about enzymes even before proteins were discovered. So catalysis is a very old story. Okay. Okay. Obviously, I forgot to mention that the sixth item here is by J. Tekamin, 1894. The first patent on an enzyme came even before we know that enzymes are proteins. Okay, so what did he do? He actually looked at an enzyme from Aspergillus oryzae. It's called takadiastase, and he patented. So here is a picture of his material that he sold, and this is an 1894 patent, US patent, where this enzyme amylase is used in starch hydrolysis, both in textile industry as well as for digestion. So very interesting background for enzymes. And today enzymes are $1 billion industry. Okay. Okay. So properly, how did people study enzymes? So obviously the reactions inside living systems, as well as some which occur outside extracellular digestion, for example. The reactions living systems require at least two conditions. One is there has to be a thermodynamic feasibility for the reaction to occur, A going to B. But just because something can happen does not mean that it will happen, this chemistry will happen. For example, glucose is stable in a bottle, but we know that glucose is a high energy molecule. It can be oxidized to carbon dioxide and water, so you can actually get a lot of energy out of it. So thermodynamically, glucose can be oxidized, but it is kinetically stable. That means the kinetic barriers have to be overcome for glucose to be completely oxidized to carbon dioxide and water. Whether you do it in one step, as in a bomb calorimeter, or you do it in a multiple steps, as in glycolysis and TCA cycle, is a different matter. Okay, but obviously, any chemical reaction potentially has two barriers. One is a thermodynamic barrier, one is a kinetic barrier. We'll spend a little bit of time on this as we go along when people study this. But emergence of catalytic phenomena, the reactions which are catalyzed, is at the core of biology. Every reaction that is catalyzed, every reaction in biology is almost always a catalyzed reaction. So we'll have some examples to look at. So this one best example I can give you is obviously many of you, even in your school days, must have looked at Haber's chemical synthesis, process of making ammonia, fertilizer industry. Okay, so where you fix atmospheric nitrogen, reduce it to ammonia. Obviously, it's a devil of a process. You see, if you want to do it, you need very high temperatures, 500 degrees centigrade. You need very high pressure, 300 atmospheres, and you need catalysts like iron. Okay, this reaction occurs. But biological systems can do such things at ambient conditions, and that is the beauty of these catalysts. I'm sure all of you know that nitrogenase exists in root nodules of the legumes, and it does the same job at room temperature, at physiological pH, but using reductants and energy in the form of ATP and NADPH and the like. Okay. But now it has become a very mild reaction. You don't have an explosion or something, but all the nitrogen fixation occurs at room temperature, ambient conditions. And that is the beauty of catalysts. Okay. Very difficult reactions otherwise can be made to occur at room temperature, physiological pH, with the kind of mild reagents that are available to you inside the cell. Okay. So that's the beauty of these catalysts. Okay. This is a slide I have taken from the Wikimedia, and all of you can see it. Maybe some of you can recognize it, that this slide has a particular order to it. Okay, this picture has an order to it. It is a simple representation of about 500 reactions inside a cell of metabolic network. And I'm sure you can recognize that the big dots are actually glycolysis followed by TCA cycle, but the small dots are all variety of steps in metabolism, and any dot is actually a metabolite. Any two dots next to each other which are connected by a line is actually a reaction between these two, the step conversion between one to the other. And obviously every two dots which are connected are right on that particular line, there is an enzyme sitting. So greater than 99% of these reactions are catalyzed. They're catalyzed reactions. So very, very rarely that you see enzymes, reactions in biological systems which are not catalyzed. Simple things like carbon dioxide, hydration of carbon dioxide and dehydration of carbon dioxide is catalyzed. The reaction is reasonably fast, but not fast enough for us to properly breathe. Okay, so you have carbonic anhydrase, which picks up carbon dioxide from muscles and converts it to bicarbonate there. And then when it goes to the lungs, you have bicarbonate goes back to carbon dioxide and lungs exhale carbon dioxide. Very, very straightforward reaction. I'm sure you must have, many of you must have looked at, CO2 dissolved in water, basically soda. Okay, it does dissolve and it does interconvert, but the reaction is not fast enough. So a primordial enzyme in biological systems is carbonic anhydrase. I'm just giving you an example. Okay, so more than 99% of the reactions in biological systems are catalyzed reactions. Okay, so enzymes are very important in that sense. Okay, so what are the features of enzymes? Okay, what makes an enzyme? The fundamental features of enzymes is that they have to perform catalysis. That means they have to hasten the reaction A going to B. Okay, in some way, they have to do this. And it's not just catalysis at random. Quite often, enzymes are very specific. They can pick up glucose in place of galactose. They can pick up, pyruvate in place of, let's say, alpha-ketoglutarate. They can, like lactate dehydrogenase versus, let's say, alpha-ketoglutarate dehydrogenase complex, or you can pick up two different amino acids required for protein synthesis. So specific by amino acid tRNA synthetase. So you have catalysis followed by specificity, two hallmarks of enzyme. And thirdly, of course, if you have a reaction which is running too fast, also is not good. You have a reaction which is uncontrolled, also is not good. So quite often, at least key enzymes, which are at the crossroads of metabolism, are very well regulated. So the three features of biological catalysts are catalysis, specificity, and regulation. You might wonder, who else has these kind of features? Okay, it turns out that antibodies can have specificity, they can be regulated, they can do binding. Okay, they can be recognized, but they have no catalysis. Okay, you can have receptors which can have very great specificity, which will do regulation-wise, signaling, but they don't have catalysis. So enzymes are the only class of biological molecules which have these three features built into them. They are beautifully crafted catalysts of nature, and they have evolved over millions of years. Okay. So, and the interesting feature is that these are functioning at physiologically mild relevant conditions. Okay. Now, what are the timescales we are talking about? So this is something which is also important, that you will see that a molecule binds another molecule, an enzyme binds a ligand, is in femtoseconds, picosecond level. But most catalysis actually occurs in millisecond range. Okay, you can see that enzyme synthesis, when you do induction, let's say in E. coli, like lacoperon, you see beta-galactosidase coming up, it might take at least half an hour. Okay, but catalytic turnovers are in millisecond range, so they're very fast, relatively fast reactions. Okay, so this is just a time scale for you to see that allosteric transitions and catalytic turnovers occur in millisecond range. Once the enzyme binds to substrate, then obviously we're talking about this. Yeah, so what I will do in the next part of the presentation is tell you something about the history of how this catalysis was understood over time and who was contributing to this. Okay, so I'll divide this into 4 parts. First, I'll tell you something about concept of catalysis and catalysts. I mean, who said what, and then how this got evolved, and then how kinetic foundations for this catalysis was established, and then what mechanistic studies were done by people, and then structure and synthesis. So this is the order in which I'll go, and each of these points I will use a few slides for that, for you to appreciate what I'm saying. Okay, first of all, concept of catalysis and catalysts. The idea of catalysis rose because of the study of enzyme action. See, this is what I probably want to re-emphasize, that catalysis was not the domain of chemists. Actually, it was the domain which chemists discovered by studying enzyme action, okay, as you can see. Hydrolysis of starch by plant amylase is similar to acid hydrolysis. This is how it was discovered, that amylase is a catalyst. Hydrolysis of meat by papain and gastric juices, I mentioned about Spallanzani, I mentioned about Riemer, mentioned about 1700s. So this is how I think the catalysis was discovered. Okay. Now, obviously, the idea of catalysis itself was introduced in 1835 by Jacob Berzelius. Okay, so the Swedish chemist and a physician. Okay, he invoked catalytic process in living organisms for the first time. Okay, so he had this to say: catalysts can rouse affinities which are otherwise sleeping at their temperature. And he also showed that these are activities, rate of reactions increase, the reactions go faster, not because of temperature. Because you heat a particular reaction also goes faster. He clearly distinguished at that time, 1835, that the effect of increase in temperature to increase the rate is different from the increase in rate due to a catalyst. And also, many of you biologists should remember that he is the first one to coin the term protein in 1838. So Jacob Berzelius made a salient contribution, the history of the first steps in understanding enzymes. Then obviously, there are always schools of thought. Then there's another chemist here, Liebig. Okay, so the catalytic force is Berzelius actually had said this in his own words: the catalytic force is reflected in the capacity that substances have by their mere presence, but not by their own reactivity. That means the catalyst is recycled to promote change in the stable molecules. So living plants, animals, thousands of catalytic processes occur within these tissues, and you will find that the enzymes actually do this job, is what he had to say. Whereas Liebig said, I mean, he had his own argument as a chemist. He had to say this, that to call a phenomenon catalytic is not enough to explain it. It is nothing but the replacement of a common word by a Greek word. So this is his argument about Liebig. Okay, and he obviously took views which are probably more chemical than a catalytic force kind of a view. But obviously, at some point of time later, you found that yeast fermentation contributes to catalysis. Obviously, Louis Pasteur showed that as a great microbiologist, immunologist. He looked at optical isomers of tartaric acid, disproved spontaneous generation life, discovered vaccination, and he proposed a vital force particular to cells. Imagine somebody who disproved spontaneous generation is now using vital force as a step to describe wine fermentation. Okay, and obviously Liebig, if you remember Liebig now, he also took up arguments with Pasteur and said chemical theory of fermentation is because vibrational impact of decomposing yeast induces sugar decomposition. It's nothing to do with vital force, is what he argued. But according to Pasteur, and he held this view till the end, that vital force unique to a living cell is responsible for fermentation of wine. Without life, those chemicals cannot be synthesized, is what he said. But obviously we know better now that yeast fermentation and catalysis was thoroughly studied by Edward Buchner in 1897. He showed that alcoholic fermentation can happen without yeast cells. So he broke open yeast cells and showed that cell-free yeast can actually ferment sugar. Reactions occur outside the living cell. Cell-free extracts can be used to make sugar get fermented. Very interestingly, for a long time, enzymes were actually called as ferments, okay, because of this. But obviously, we know that we call them enzymes later. Okay, so Buckner's contribution was seminal in a sense that he showed that you don't need a living cell, and you can still do conversions. So obviously, he disproved Pasteur's vital force theory, and he also showed that you can actually have enzymes work in cell-free extracts. You must notice that working in cell-free extracts for any catalyst is a big thing. Why is it a big thing? Because once you show that a cell is not required, you can now start splitting this extract into components and isolate individual components. That means you could isolate all the enzymes of glycolysis. And that was the basis of how glycolytic enzymes were discovered. So Edward Buckner's contribution to enzymology is enormous, okay, from that point of view. Obviously, Buckner was part of a Adolf von Baer School of Chemists in Germany. And one of his colleagues was Will Statter, who's also a chemist. We'll hear about him a little later. But what obviously Adolf von Baer said, that because this guy discovered cell-free extract do fermentation, this will make him famous, even though he's not a great chemist. He has no talent as a chemist. So this is exactly the comment made by Buckner's guide, mentor himself, okay, as quoted by his colleague, Will Statter. So, but you appreciate the fact that chemistry has contributed. Bayer School actually is a chemist school from Germany, has contributed greatly to biology, greatly to enzymology. I'll show you in the next slide. It's a very busy slide, but you will obviously realize that Adolf von Baer, good scientists generate good scientists. Okay, his school had 4 major contributors to the field of chemistry and biology: Will Statter, Wieland, Fischer, Buckner. Buckner is the one who actually contributed to the yeast fermentation and showed that cell-free extract can do fermentation. Okay. Whereas the others, Will Statter and Kohn, Wieland and Linen, okay, and obviously Fischer's school was very big. You can see a lot of people coming out of that. And this whole picture that I'm showing you has a color code in it. The guys who are in red are the guys who contributed both to chemistry and biology. And the guys who are in black are excellent chemists, organic chemists. The guys who are in blue are actually enzymologists and biologists. I'm sure you will recognize names like Krebs, Thierel, Severo Ochoa, Lippmann, Meyerhoff, okay, and Fedor Lenin. So many of these people contributed to enzymology and study of metabolism in some sense. What is common to all this is Bayer's school. These are the scientists, all of them are Nobel laureates. Okay, so they have contributed to this field of interdisciplinary research. Okay, so Buckner's work showed that enzymes are catalysts, and enzymes are there in the cell-free extracts, which killed the vitalism theory of Emil Fischer, and obviously the birth of enzymes came about. Whether it has one enzyme or many enzymes is a different matter. With time, we try to resolve every one of them, and now we know there is glycolysis, there is a TCA cycle, and a whole lot of things. Okay, obviously around that time, chemical synthesis of urea by Wohler, 1828, from ammonium cyanide dealt the final blow to vitalism. So that's gone. So I think study of enzymes, in some sense, actually allowing to study enzymes killed vitalism, theory of vitalism, because enzymes now allowed you to show and do many reactions which were otherwise thought only done by living beings. So you could do these reactions outside a living being, is exactly what it showed. And in fact, the emergence of modern biochemistry was in a large measure the gradual replacement of the vital force theory by the use of enzymes and study of enzymes. Okay, so what actually enzyme means? Enzyme actually rightfully means in yeast. Okay, the term enzyme was coined by Wilhelm Friedrich Kuhn in 1867, and I'm sure you will realize why it is used that way, because most of the study of enzymes came through the study of yeast. So they were, and first time the enzymes were studied from yeast extracts. So in yeast, yeast zyme. Yeast is called zyme. In yeast is actually enzyme, and its activities are resolved through the prisms of biology, and a lot of chemists contributed to it to create the world of enzyme, word enzyme itself. Okay, so that I kind of gave you a background about how catalysis and catalyst came about. Okay, who are the people involved? Okay, in understanding this. Now let me go to how people studied kinetics, because any reaction is studied by the product formed, the nature of the product formed, the stoichiometry, and how quickly it is formed, the rate of its formation. So all this involves kinetics. And the kinetic foundations for enzymology were also laid out pretty early. So we will look at how they were done over time. Okay, that's what we know today. Yeah. So then we'll go to the remaining two. The kinetic foundations is also laid by Pierre Berthelot. Berthelot again was a chemist, was the first to derive a second order rate equation. Okay. And in fact, this is where most biologists start wondering whether I'm going to derive a rate equation. No, don't bother about derive a rate equation. Most rate equations have already been derived. All you can do is use them if you know how to use them. Okay, so that's all that is required. So Berthelot derived the first time the second order rate equation. And what is important to remember today is that in most enzymology that we do today, you don't even need a second order rate equation. You most of the time need first or zero order rate equation. Okay, so we will come to that in a minute. Okay, so Berthelot work on second order rate equation eventually influenced Goldberg and Wall's law of mass action, and that came to chemical kinetics. Okay. And in fact, Berthelot extracted invertase, again a ferment, from the yeast. Invertase is an enzyme which converts sucrose to glucose and fructose from yeast, and studied it. So a lot of his kinetics was done using invertase. So again, yeast enzymes became very important in laying foundations for kinetics as well. Okay, so this enzyme invertase, what was shown is that it exhibits what is called as a saturation kinetics. If you follow the rate of the reaction as a function of its substrate like sucrose, okay, if it is a reaction which is hydrolysis by, let's say, simply acid catalyzed heat reaction, uncatalyzed reaction, then you will get a first order linear slope of 1, for example, okay, uncatalyzed reaction. But the moment you have an enzyme, you have a catalyst, you would start seeing a saturation, phenomenon of saturation. You'll reach a maximum rate as you increase the concentration of the reactant substrate. So catalyzed reactions show this kind of a behavior, particularly substrate saturation, particularly maximal rates. Okay, and as you can see that in the beginning, the rate of the reaction is first order. At the end, the rate of the reaction is independent of the reactant concentration. Okay, that means it's a zero order. Okay, so this is the range in which you go from first order to zero order. Okay, x raised to 1 to x raised to 0. And obviously, even if you increase the concentration further, there is no increase in the rate. Whereas uncatalyzed reactions, you keep on increasing the concentration of the reactant, rate keep on increasing linearly. Okay, that's the big difference. So this is how it was understood in the first place, using invertase as a model enzyme catalyst. Okay, so using invertase again, Victor Henry for the first time derived the hyperbolic rate equation to define this hyperbolic curve, the catalyzed reaction curve in 1903. Obviously, many of us, when we look at enzyme kinetics, we come across this Michaelis-Menten equation, which was derived in 1913. Okay, but you should appreciate the fact that one form of this equation was known 10 years ago. So a lot of people contributed to this development, and obviously, rightfully, this equation should be called Henry Michaelis-Menten equation. What is interesting about this equation, as I've shown here, is Henry Michaelis and Maud Menten, the first enzymologist lady, they actually derived this equation, and 2013 was the centenary of this equation derivation. Okay, whole books, whole issues of biochemistry were actually dedicated to this. So what is interesting to note here is that if you look at this equation, and I want you to do a very quick trick on that, if S is very large, okay, so that Ks can be neglected, then Ks plus S becomes S, and you can cancel off S, B becomes B max, right? So that means there is no effect of substrate. If you look at that, that is when you see X equal to, zero order rate here, so S is very large, so it becomes a constant. But if S is very, very small, then Ks becomes important. Ks plus S becomes Ks itself. So then it becomes a rate constant followed by a substrate concentration. It is the first order. So in fact, this equation beautifully describes the behavior of this enzyme saturation. And without exception, all enzymes with their substrates behave in one or the other way related to this kind of a saturation. Okay, so it's a very fundamental discovery. Not much has changed after that. Okay, of course, JBS Haldane contributed to this work, but this is what I want to tell you, that this equation came about in 1913, and it is being used heavily even today. Various forms of this equation are used even today. Okay, so the another contribution that came about to study enzyme catalysis is what is called as, in chemistry, people looked at transition state theory. So that developed in chemistry first, but it was very quickly applied to enzyme reactions. So what is transition state theory? It tells you that a reactant, okay, if it has to go to a product, there is an activation energy barrier. So you have to provide some energy to cross that barrier to give you the product in the reaction coordinate. Okay, what a catalyst does is it decreases this barrier, okay, so that the reaction go faster. So Arrhenius looked at activation energy, and eventually people showed that enzyme catalysis also follows the same principles, that it indeed decreases the rate activation energy and hence increases the rate of the reaction. So enzymes are in that sense catalysts. And what you notice is that substrate S from left going to P, this is downhill because P is at a lower energy level. So this is an exergonic reaction. That means the reaction gives out energy. So it is like glucose oxidation. But if you want to make glucose from carbonate and water, P going to S, you have to also input energy. Think of photosynthesis, for example. Okay, in both cases, you need an activation energy barrier to be overcome. So whether a reaction is thermodynamically feasible, okay, or kinetically feasible or not, as long as you have a catalyst, you can make that reaction go. If it is thermodynamically not feasible, you can provide energy and make it go. If it is thermodynamically feasible, you can only provide a catalyst and make it go. Either ways, catalysts are very important. Either ways, catalysts are very important. And these catalysts in biological systems are nothing but enzymes. Okay, so this was also adopted very quickly to biological systems. And I'm sure you are all familiar with Linus Pauling. Okay, and once people thought that transition state level itself is brought down, activation energy level is brought down by catalysts, people wondered how it can happen. Okay, and one of the concepts was by Linus Pauling. He said, I think enzymes are molecules that are complementary in structure to the activated complexes of the reactions that they catalyze. Just appreciate what insight this man had, okay, this great scientist had. We all think that enzyme binds substrate. Enzyme might bind product, maybe poorly. But what he said, if enzyme bound substrate, it would just like be an antibody, right? It will be more like a receptor binding a ligand. But enzyme does not bind the substrate, nor does it bind the product so well, but it binds the transition state better, so that it brings down the level of the energy level of the transition state. So enzyme bound substrate is actually taking the shape of a transition state, and hence the reaction is accelerated. So this is what he said. And obviously you see a direct implication of this. If you could make a molecule which is like transition state, and you raise an antibody against the molecule like a transition state, that should be able to be a catalyst. And in fact, we know that today because people have created enzymes like catalytic antibodies, where you can take a transition state like structure, raise antibody against them, and use these antibodies to do the catalysis. So there are at least 5 or 10 enzymes in the market today for reactions which were not normal, because you can now think of any reaction for which you can raise a catalytic antibody in principle. Okay, so this has been done. So you can imagine 1948, this was what was proposed by Pauling, and it was eventually confirmed. Obviously, Pauling is well known for alpha helix, protein structure, and many things. Okay, so I told you something about kinetic foundations, okay, how kinetics, saturation was discovered. Now, also look at mechanistic studies. What kind of mechanism that enzyme goes through for catalysis was also studied. Again, Emil Fischer, the first Nobel laureate that I showed you in the list. So he looked at a number of systems, particularly sugar molecules with different stereochemistry, and he showed the importance of stereochemistry for physiology in 1898. Okay, based on their sugar specificity, intuitively proposed that enzymes actually bind one kind of sugar or the other, and he said implicitly the enzyme has to bind one sugar but not the other. So he brought about the concept of lock and key, okay, lock and key concept. The fact that catalysis can occur, that means an enzyme should bind the substrate or a ligand, implicitly means that the complex has to form. So mechanistically, a reaction catalyzed by an enzyme cannot happen at a distance. It has to happen on the enzyme. That means enzyme substrate complex have to come together, then only the reaction can proceed. So this is a very fundamental concept, because if the substrate by itself was doing it, then there are implications. But if the product is coming out of an ES complex, there are other implications. I'll show you that in a minute. For example, invertase, again, example of invertase from yeast, invertase sucrose complex. Okay, A.J. Brown in 1902 showed that formation of an enzyme substrate complex does occur. He deduced it from substrate saturation kinetics. The same data can be used. See, if the substrate was going to the product, okay, without the enzyme directly interacting, then obviously there is no reason why you see a saturation phenomenon. But if the moment you see a saturation phenomenon in enzyme catalysis, you can say that enzyme substrate complex is formed and the product comes out only of the complex and not out of the substrate. So even if you keep on increasing the substrate, what limits is the extent of enzyme substrate complex. So enzyme is limiting, substrate is in excess, ES complex is limiting. So you will see a limiting rate. So a phenomenon of substrate saturation kinetics actually tells us that there is a ES complex form. Okay, so this is the kind of analysis people did pretty early, almost a century ago, to propose that enzyme substrate complex is a prerequisite for the reaction catalysis to occur. Okay, so then again, questions come in terms of what really are enzymes made up of. People did not know for a long time. Okay, in fact, even in 1920s, people did not know what is an enzyme. Okay, so Willstatter in 1922 proposed what is called the trigger or carrier theory. Enzymes contain smaller reactive groups. Because he was a chemist, he thought there has to be some reactive group attached to an inert colloidal carrier, and that is a catalyst, is what he proposed. But then Sumner, I'm sure all of you know, is the first one to crystallize urease and show that urease crystals are protein. Okay, so he said enzymes are proteins in 1930. In fact, he got his Nobel Prize for that. The first one to crystallize an enzyme and show that all enzymes are proteins. Okay, so this is the argument again, tussle between two great scientists. A little bit of a background, J.B. Sumner did a lot of purification of this urease enzyme. Okay, and particularly for the younger audience, I want to tell you that Sumner wanted to do research, but because of an accident, he lost his right hand. Okay, and no guide wanted to take him, because he would not be able to work in the laboratory. But with great difficulty, and he did a lot of work with single-handedly. He did crystallization and purification, and he was rewarded for that. So it's a great true story to see how Sumner actually overcame a lot of difficulties to study enzymes, and contribute to this field of enzymology. Okay. So this argument between Willstatter and Sumner was so bad that Sumner was in Yale University in US, whereas Willstatter obviously was a Bayer school. He was in Germany, so he did not like Sumner's ideas. So he went all over to the United States, to Yale, and he trashed him in his own university, because Willstatter had such a big stature, so he was able to say and get away with it. But we know today that Willstatter was wrong. Okay, so obviously Sumner showed that urease crystals were actually protein. Okay, but let me not jump to the conclusion. I'll show you the next one and see how interesting things are actually in science. Okay, so this protein nature of the enzyme, what was the argument for and against? Willstatter and Sumner. So Willstatter started using a reaction of an enzyme called peroxidase as his model, whereas Sumner used a reaction of an enzyme called urease. Okay, now in those days the chromatography was just building up. People did a lot of chromatography, built a lot of fractionation techniques. Sumner used ammonium sulfate acetone precipitation, got jack bean urease, pure crystals, and in those days, I'm sure many of you know that protein estimation and protein detection, how is it done today? It is done by Bradford method. Before that, it was done by Laurie's method. And before that, it was done by biuret method. In fact, in those days, it was only biuret method. So if you needed a large amount of protein to actually show that it is a protein, okay? And in fact, Sumner had good crystals, a lot of protein, so his protein test came positive. Whereas Will Statter actually purified this enzyme peroxidase from horseradish using C-gamma gel, binding and dilution. He got a fraction, but it was in such small quantities, it came out protein test negative. So self-fulfilling prophecy, Will Statter said enzymes cannot be proteins, but Sumner said enzymes have to be proteins. Okay, but eventually it turns out that when you have more sensitive method of detection, even peroxidase is a protein. Right? So we now today know that all enzymes are proteins. Obviously from Zek's work, we know that some ribonucleic acid can also be a catalyst. But what is interesting to look at is we say all enzymes are proteins, but then ribosomes came, Altman and Zek. Ironically, if today Will Statter and Sumner had argued about this, probably Will Statter would have still been right. You know why? Because in those days, when Sumner actually purified urease and he showed protein test positive, if they had done a nickel test positive, that also would have come positive because urease is a nickel enzyme. And then obviously Will Statter would have said, look, there is a reactive group attached to inert colloidal carrier. That's why it is an enzyme. But fortunately, sometimes it's good that you don't know everything, that it turns out that nickel could not be detected. So urease was considered as only a pure protein, and hence obviously Sumner and the whole field moved into all enzymes are proteins. Okay, very interesting story actually. As you can see today, that urease catalyzes the reaction, urea plus water giving you ammonia and carbon dioxide at this active site actually has 2 nickels. It's a binuclear nickel center. So if you look at urease today, you will definitely say that there is an active site, there is a reactive center. Okay, and obviously Will Statter would be right. So quite often, as it happens in science and biology, that there are many things that people say, but the truth is somewhere in between quite often. So it is true that there is a carrier, it's true that there is an active site. So both were right in their own sense. Obviously their models were different. So today we know that enzymes do have cofactors, okay, to work. And metalines are some of the kinds of cofactors that we see in enzymes. Okay. So enzyme history was a journey of many ups and downs, typically of any scientific progress. Okay, a complicated hypothesis is proposed, which usually entails an element of mystery, and several unnecessary assumptions are made. This is opposed by a more simple explanation, which contains no unnecessary assumptions. The complicated one is always the popular one at the first, but the simpler one, as a rule, eventually is found to be correct. This process frequently requires many, many, many years. Okay, and people do have, as you can see, have to wait for recognition for a long time before their discoveries are actually validated. That's what Max Planck said. In such situations, scientists never change their minds, but eventually they die. Okay, so you will see that some of the contributions made by scientists, particularly in fields which are very competitive, get accepted much, much later. Enzymology is an example of that. Okay, so the last bit I want to cover as a part of this presentation is structure and synthesis. Okay, obviously, enzymes were treated by chemists as natural products, and chemists always went through the study of any natural product, like isolated structure, then do total synthesis, typically how natural products were studied. So study of enzymes also has the same stamp like a natural product. So people isolated the enzyme, they did structure isolation, and they eventually did total synthesis. So I'll show you structure and synthesis. Obviously, first thing people did was to sequence these proteins. Okay. Ribonuclease was the first lucky one to get sequenced. Obviously, insulin is not an enzyme, but it was the first protein to be sequenced. But ribonuclease is the first enzyme to be sequenced. And the structure, as far as structure was concerned, myoglobin was first to be elucidated. But obviously, the first enzyme was lysozyme, was elucidated, structure was elucidated. And obviously, the first enzyme whose total synthesis was achieved was ribonuclease A. Sure, you know this. Okay, so here is the total synthesis of ribonuclease A example. There are 124 amino acids, so you could do a whole range of synthesis using solid phase peptide synthesis. And Bruce Merrifield got a Nobel Prize for this work. Okay, and now it's mighty complicated. We take it for granted that 124 amino acids are attached one after the other on the ribosomes, and you get a synthesis. So synthesis occurs from N to C terminus. But when you have so many different amino acids and so many functional groups, it's not easy to do synthesis. So a lot of blocking, deblocking, a lot of functional chemistry got done, and solid phase peptide synthesis got done, and a functionally active enzyme of ribonuclease was actually synthesized on a solid phase peptide synthesizer by Bruce Merrifield. Today we can synthesize a number of peptides and small proteins at our will. Okay, peptide synthesizers are available. Okay, so this is how it works. Synthesis was done. Okay, and one of the more recent areas in enzymology is actually what's called structural enzymology. Okay, we would like to understand how enzyme functions, so we need to look at the structure. It is particularly interesting for those enzymes where, you know, not everything inside the enzyme is so easily available as a readout in terms of its catalysis. For example, okay, this is something which is interesting. There are enzymes where there are protein tunnels inside an enzyme. So these tunnels are used for moving small molecules like indole, ammonia, carbon monoxide. These are reactive toxic molecules sometimes, which are part of the reaction. They are not let out of the enzyme, but they are actually moved inside the enzyme itself. And such tunnels cannot be seen by simple kinetics or simple study. You need to look at the structure to understand the tunnel. So I'll show you an example of one enzyme here, which is tryptophan synthase. So tryptophan synthase catalyzes a reaction where indole glycerol phosphate is converted to tryptophan by its reaction with serine. Okay, but the indole glycerol phosphate actually releases glycerol 3-phosphate, and then indole formed does not come out of the enzyme. As you can see in the picture, there is a tunnel of 40 angstroms long, where the indole glycerol phosphate is on the top, IPP, which is actually an analog, but that is where the indole glycerol phosphate binds, and then the glycerol 3-phosphate leaves, whereas the indole does not come out. It just goes through the tunnel, walks about 40 angstroms, and on the other end, it meets serine, and actually tryptophan is formed. Very interesting. So why is it required? Because indole is toxic, so you cannot have it out. So this two-step reaction is done through a tunnel like this in a single enzyme. Okay, so there are many enzymes like this where there are tunnels where the active reactive molecules or intermediates go through. Okay, and this kind of a picture you will not get unless you have a structure of the molecule. So one of the contributions for crystallographers and structural people, biologists, is they are able to show this kind of mechanistic details of how catalysts work, how enzymes work. Okay, so this is an interesting example. Of course, there are many other cases. So as a consequence of this, what happens is a molecule goes from one place to another without actually coming out of the enzyme or coming out of two enzymes. Basically, you can view this as two enzymes, one reaction on one end, other reaction on the other end. So there are many examples like this. And in fact, in addition to protecting the cell, this kind of machinery is also useful in holding very important resources within instead of letting them go. So there are a number of things that happen inside a series of reactions inside a cell where you need to protect these intermediates. So there are regulatory features which involve enzymes which have evolved over time in nature. And I'll show you some examples of that here as an extension of this discussion. Okay, so the top picture here on the left is actually a molecular tunnel. I showed you already, but obviously you must have realized that in fatty acid synthesis, or in the case of, let's say, pyruvate decarboxylation, pyruvate dehydrogenase complex, the intermediates are actually shepherded, just like a molecule on a leash, taken from one active site to the other. And obviously in some systems where there is neither of these, neither a tunnel nor an intermediate, but actually two enzymes come together, they are neck to neck with each other. So the intermediate does not fall out. Okay, this is another way of trying to handle conjugative steps in metabolism, and obviously this is part of regulation. Okay, on the right top corner is where normally reactions, the way enzymes perform, is substrate goes to the enzyme, product X comes out, it is taken by the next enzyme, goes to the second product. So if there is no channeling, there is no tunneling, there is no intermediate, there is no metabolon, then obviously you see a very simple system. But today, I think what we are also seeing is that some enzymes in large concentrations, some enzymes in small concentrations inside the cell. So there is an aggregate clustering of these enzymes occur, and the enzyme in the center here shown at the bottom right enzyme in the center, actually releases a product which has no way of escaping but to hit the second enzyme and finish off. So the reaction has to complete. So these are kinds of things people are discovering, how enzymes are regulated inside the cell. There are many other ways of regulation, but this is one way I'm trying to show you where structure also matters in trying to do the regulation of the enzyme reaction. Okay. So what is the future that we have for the enzymology as of today? If everything is known, or do we know everything, or we still have a lot to learn? I think there is quite a bit to learn. Okay. At the molecular level, we want to understand how enzymes work. Is we need to look at transition states, how actually this transition state is formed, and can we understand it? A lot of computational enzymology is going on, computational work is going on in this. This is one frontier. Okay, for almost any enzyme, we should be able to generate a transition state structure. Okay, which means we can generate a catalytic antibody, which means we can make an enzyme de novo. So there is also a lot of work which is going on, and people are contributing a lot in terms of structure function dissection of enzyme catalysis, which includes site adherogenesis, crystal structures, enzyme allostery, single molecules studied as a single molecule or also inside a cell. We can also look at enzymes whose structures can be predicted, and then we can build those enzymes or use enzymes to make fresh enzymes. So the latest and the last I want to share with you is enzymes can be made to order. This is the ultimate that we want to do. So if I have a reaction A going to B, there is no enzyme available in nature, can I make an enzyme? Okay, how do I make this enzyme? Okay, is it possible? I think that is the challenge, ultimate challenge to how to study enzymes, okay, how to work with enzymes. So we work with catalytic antibodies, that's one way, ribozymes, one other way, but they are not as good catalysts. So there are people who are working on designing enzymes from the protein as well as non-protein scaffolds who make catalysts. Okay. But as Thomas Edison said, until man duplicates a blade of grass, nature will laugh at his so-called scientific knowledge. Unless we make an enzyme from scratch, we will still not have understood everything about enzymes. Okay, so I thought I'll mention that. Obviously, we also look at future of enzymology inside a cell. Inside a cell, what are the things that contribute to a function of an enzyme? Let's say metabolite A goes to metabolite B. We have an enzyme. Okay, it's not a single enzyme. In this case, it is a single enzyme, but if there are a lot of metabolites, there will be a lot of enzymes. But look at a single enzyme, its rate of the reaction, velocity is affected by such features like Kcat, Km, NH, E total amount, number of molecules, substrate concentration, inhibitor concentration, activity concentration. So the readout of this reaction, A going to B, is the function of all of this. Okay. So it's a very complex system. And this is for one enzyme. Inside a cell, there could be so many ways you can regulate the enzyme activity. But if you have a number of enzymes which are working together like this, then obviously the regulation becomes a much more complicated event. So one is looking at in vivo enzymology also as a possible frontier in study of enzymes. So concluding, the study of history is an exercise in humility. Enzymology is no exception. History of enzyme catalysis is no exception. Some of the best minds work in this domain of science at the intersection between chemistry and biology. And obviously, there is a lot to go to understand in the future. I think we'll stop here. I can take some questions. Thank you. Yeah? Hello?
Praveen sir,
Yeah.
can you please read the questions for sir?
Praveen, are you doing it?
Himanshu sir. Praveen sir,
Yeah, madam.
Madam,
Yes,
can you read the questions in the chat box?
Yeah. Yes, madam.
Yeah, please.
Yeah. Good afternoon, sir.
Yeah.
Yeah,
Right.
there are some few questions in the chat box. I'll just ask one. Sir, Miss Jaya wants to ask. There are two questions proposed by her.
Yeah.
One is, what drives the movement of reactants or molecules in a tunnel in tunnel enzymes from
Yeah.
one side to another?
Like everything else, it is concentration. See, if you put a particular molecule, let's say you take a beaker, okay? You put a crystal of sugar in this water, okay? Obviously, the crystal of sugar will be at one place, and surrounding will start dissolving in water. It will diffuse out, right? You'll set up a gradient, okay? So similarly, wherever there is a source of a material, obviously where there is less of that, it will move in that direction. It's just entropy, right?
Okay.
From a source to a sink, or from a higher concentration to a lower concentration, because constantly from one end it is being withdrawn, it's being removed as a product. So you make at one end, you take it. So it's, I would say, example of a molecule diffusing in water is a three-dimensional diffusion, okay? Whereas a molecule of a lipid diffusing in a membrane is a two-dimensional diffusion, only two directions, X and Y. But in a tunnel, it is a one-dimensional diffusion. Okay, so you have a three-dimensional diffusion, you have a two-dimensional diffusion, you have a one-dimensional diffusion. In fact, people believe that that one-dimensional diffusion provides actually an exciting way of increasing the rates of the reaction, because the molecule does not go left and right or anywhere, goes in only one direction. So the rates are much more because of a tunnel. Yeah.
Yeah. Sir, in addition to that, she asked why such tunnels have evolved.
Okay, I mentioned that, but since I was doing other things in trying to cover, I didn't say this. Look at indole, like tryptophan synthase example I gave. Okay, so do you know what indole is? Indole is an intermediate in this reaction, but it does not come out. And I'll tell you that indole is a very toxic molecule. No organism will have indole, but all organisms will have tryptophan. The reason is that if you have indole in solution, it's toxic. So one reason why these tunnels are made is that the intermediate is toxic to make sure that it does not go out. It quickly gets captured. Yeah. So you make indole and you use it right away. It's more like a very, very corrosive reactant, right? So you don't want it to be going out. That is one reason. I'll tell you the other example. Carbon monoxide, for example. Okay. So methanogens have these enzymes where carbon monoxide is fixed. Okay. Carbon monoxide, once it is in the gaseous form, it's very difficult to capture it. So it gets made and channeled through a particular form. So you are locally increasing the concentration. That is one way of doing it. The third example I gave you was of ammonia, right? There are ammonia tunnels. I'll give you an example of an enzyme where ammonium is required. Okay, ammonium is required. So I put ammonia in water and make this reaction happen. It will not happen. Why will it not happen? Because in solution, under physiological conditions, ammonia is present as ammonium, NH4 plus. Okay, but NH4 plus is not a good nucleophile. If you understand a bit of chemistry, it does not 2 electrons. It's already NH4 plus, right? All the bonds are satisfied. But if you remove a proton from there, then it becomes NH3 with a lone pair. Okay, this is a lone pair of electrons. And this lone pair is required for the chemistry. So in water, if you want to put ammonium, it's called nascent ammonium. So you must have heard of in school chemistry, nascent chlorine. It's not Cl2, it is Cl. Similarly, you have nascent ammonia released, which is a good reactant for the chemistry to occur. But how do you release nascent ammonia? You cannot take ammonium and make it ammonia. Not so easy. Not so easy at all. So what nature does is that in such enzymes, it takes glutamine. You know what glutamine is, right? Amide of glutamic acid. Okay. And it uses glutamine, it hydrolyzes glutamine in the active site to create ammonia, not ammonium. Okay. So this ammonia is not even let outside. It is captured in a tunnel and pushed to the other step. So a number of reactions like this, where the reagent has to be, protected from the solvent, you will find enzymes have tunnels. Is that okay?
Yes.
Yeah.
So there's a question by Ms. Tanya.
Okay.
Are molecular chaperones also enzymes?
They are. Basically, the molecular chaperones have a cavity where they can take the unfolded molecule of a protein, okay, and at the expense of ATP, they can allow you to sample all the possible conformations of the molecule, so that from an unfolded to a folded state, there's an equilibrium established. Whichever is the folded state is often the most stable state. Once it is achieved, it is put out. So chaperones do have an ATPase activity. I hope you understand that. No, they have to input energy to be able to unfold a molecule first. But only folded molecule also has to be unfolded before it can be refolded. Yeah.
So there's another question from Mr. Bhagirath Mishra. Sir, can you please explain what is present in active site of ribozymes?
Ribozymes. Okay. I should tell you something about what is at the active site of a protein enzyme in the first place. Protein enzymes. Protein enzyme active sites have amino acid side chains, particularly the ionizable side chains, and also the polar side chains, like chymotrypsin has serine at the active site. Okay, and chymotrypsin also has a histidine at the active site, side chain of histidine at imidazole at the active site. So you will find many of these side chains at the active site in a regular protein enzyme. And sometimes if these regular side chains of 20 amino acids are not sufficient, not enough for the chemistry, then the nature has recruited cofactors like pyridoxal phosphate, like a metal ion, like in case of urease. So a combination of a protein and a cofactor will make sure that a catalyst functions, right? This is how it is. Now you compare this with the ribozyme. So what would ribozyme have? What kind of site, what kind of reactivities ribosome active site would have? Okay. So obviously it is a ribonucleic acid. Okay. It has sugar OH groups, some of them, not all of them. Okay. It has phosphates. It has bases. Okay. Like purine bases and pyrimidine bases. And even in these bases, they have NH2 groups. Okay. So when the ribozyme folds itself, okay, into a functionally active molecule, you will see that some of these groups are pointing towards the active site. So they perform the catalysis, they're part of the catalysis. And obviously ribonucleic acids are not naked molecules, they are always coming with magnesium. You see, they are complexes of magnesium complexes, manganese complexes. I'm sure you know that all the RNA and DNA have counter ions to them, and magnesium is the most common counter ion that is possible. So you'll find that ribozyme will have magnesium bound to it. Sometimes that also will be participating in catalysis. Is that clear?
Yes, sir.
Yeah.
So Kale sir is asking, sir.
Yeah, sure.
Kale sir is asking, do the stress proteins catalyze reactions, or are they only messenger?
Which ones? Stress, say it again.
Stress proteins.
Stress proteins. If one can be more specific, no, because a stress response is also chaperones in some sense. Chaperones also do their levels go up when there is stress, right? Chaperone levels go up. Okay, some of the stress proteins, obviously, it depends on the kind of stress. Okay, if there is a nutritional stress, obviously there are a number of kinases are involved. TOR signaling, for example, is part of nutrient signaling. Okay, so it is not necessary that every molecule in the stress response is a catalyst. Okay, there could be receptors. There could be molecules which will activate a particular enzyme which already exists in a, let's say, less active form. So these are possible.
Okay, sir.
Yeah.
This is another question. Ms. Sangeeta asks, how do we calculate the zero order, first order, and second order reaction in the numericals?
Numericals. There are standard formula for that. Okay. Basically, a zero order reaction is one where there is no dependency, on the concentration of the reactant. So the reaction will proceed regardless of what concentration of the reactant you use at the same rate. Okay, it's a fixed. Whereas if you look at a reaction where the concentration to the power 1, is involved, then obviously it's a first order reaction. Okay, so rate of the reaction is equal to rate constant into the concentration of the reactant. That's typically the first order. Okay, but if it is a second order reaction, then obviously rate of the reaction is equal to a constant, which is a second order rate constant, followed by either the concentration squared of a single molecule, kind of a molecule, or two different molecules, two substrates, A and B. So then also there will be two concentrations, right? So two concentration terms, and this is how you will see the simple presentation of the reaction rate equation. Okay, this can further be analyzed and derivatized to get you a state where you can do a functional rate measurements. Because in rate measurements, you don't have one concentration. You use a number of concentrations to study it, okay, to develop the entire rate equation. Yeah?
Yes, sir.
Yeah.
So there is one more question.
Sure.
From Subhash Tripathi.
Mm-hmm.
What makes tau polymerase resistant to heat, whereas other enzymes are thermolabile? Not TAC, TAC
Naveen said
polymerase.
TAC.
Okay. So I think this is one area where I think a lot of good study of enzymes is going on today. Okay, where people are looking at exotic ecological niches like the deep sea vents, thermal, geysers, and wherever there is unusual kind of psychrophilic organisms in very cold conditions. Okay, so the function of an enzyme, we should understand, is threefold. I mentioned, right? So first of all, it has to be a catalyst. Secondly, it should have specificity. Thirdly, it should also be potentially regulatable. Forget regulation for a minute. It should have specificity. It should have ability to catalyze. And since it can bind the substrate and it can bind the product, substrate is related to the product, okay, in some sense. Because let's say we talk about glucose isomerase. It should bind glucose, it should produce fructose, or it should bind fructose and it should produce glucose, right? So glucose isomerase can bind either glucose or fructose. They are related to each other, but they're not same. Okay, so that means the enzyme active site should be flexible enough to accommodate at least two structures. Okay, if there are enzymes with more than one substrate, then obviously it's further more complicated. So what I'm trying to drive at is that the enzyme active site is not a very rigid entity. It is a very dynamic entity. Okay, there is a lot of conformational flexibility. Okay, and it may be local at the active site, or it may be global. Okay, it may be all over the molecule. Okay, so it depends on what kind of function that enzyme performs. The flexibility of the molecule is determined by that, and nature has evolved that flexibility. Okay, now if you have an enzyme which has to function at a particular temperature, okay, higher temperature, there is a lot more thermal motion, right? So weak interactions which hold two, let's say, two helices together or two parts of the protein together, okay, are easily broken. All right. So whereas if you have very cold temperatures like Arctic and Antarctic fish enzymes in them, then obviously it's very low temperature, so there is very thermal motion is less. So it's not very easy to break open a molecule. So the kind of interactions within the enzyme molecule to hold it into an active structure. Active structure is a dynamic structure. It's not a frozen structure. So a whole range of thermodynamic considerations of how the side chains interact, how much of flexibility in the molecule exists, all these matter. Okay. Now, one way people are trying to make an enzyme more stable, for example, is they look at the structure and see that two parts of the molecule fall apart when the temperature goes up. Okay, let's say a mesophilic enzyme which works well at, let's say, 37 degrees centigrade. I want to make it to a thermophilic enzyme, which is a very stable enzyme at higher temperatures. What happens is this enzyme is taken to higher temperatures, it starts opening up. The molecule starts opening up in terms of a protein structure. Okay, but if there are regions in the enzyme which are more susceptible to it, and suppose if there are amino acids next to each other, where I can do replacements and probably build a disulfide bond. Okay, I can build a disulfide bond so that this disulfide bond is a covalent bond. Now the molecule cannot fall apart. So one strategy is to build such knots in the molecule so the molecule becomes rigid. But you must understand that such rigidity in the molecule might also have an effect on its ability to do good catalysis, because flexibility is important for catalysis, right? So I think one of the challenges today is to be able to figure out for each enzyme molecule of interest, like Taq polymerase, for example, what makes it so stable? What are the forces, intramolecular forces that make it stable? And if we understand that, we can build other molecules like that. One of the things people are doing is introduce disulfide bonds to make a molecule more stable. Okay. The other thing that people are trying to do is to be able to introduce, interactions between two parts of the same molecule through, let's say, a more hydrophobic interaction than, let's say, hydrogen bonding or salt bridges and things like that. So it depends on case basis how exactly you want to build the strength of a three-dimensional structure of a molecule. Is that clear?
Yes, sir.
Yeah.
Sir, that was the last question.
Okay. Yeah.
Yeah, there are a number of compliments for you for a very nice and lucid lecture.
Yeah.
Yeah.
Okay.
Yes, sir.
I just wanted to say this, that my experience of teaching this subject for so many years, I realized that biology students are very quickly put off by all the mathematical jargon and equations in this. There is actually not so much of this jargon. If you understand it well, it's not required to have too much of math. In fact, what I would say is that you need one or two basic algebra equations, and rest of it is all your imagination, how well you can understand the system. Okay. It's not really that much of mathematics. Of course, you can do a lot of math if you want, but it's not required. For doing good enzymology, routine enzymology for studying biological systems, you don't need so much of math. So I just want to tell all those younger audience who probably attended this talk that enzymology is a subject which is taught in a very dry manner. It is not that dry. Look at history of how enzymes came about. You will appreciate a lot of things. Why something is hyperbolic? Not because your teacher told you, because it is hyperbolic. It's hyperbolic because the way the system actually has evolved. Yeah. Okay.
Yes, ma'am.
Yeah.
Madam?
Yes.
Yeah, question answer session.
Sir, history is something I think each student should know for every topic, because it also paves a way to think and go forward.
Yeah.
And you narrated it so well that I'm very sure many of us who have been teaching some of these aspects are rethinking now, that we can revisit that entire topic and give it a different dimension. So absolutely.
Actually, my own reading is, if you give examples from history, students tend to forget less. If you tell them as only a topic, as a concept, then they tend to forget because they don't see a value for it immediately. But if I tell you that, look, this is how it went, people fought about this, then eventually it got discovered like this, they cannot forget
storytelling.
Yeah. They don't forget the background.
Yes.
That's very important.
Amazing. It was too good, sir. Too good. But you also put flavors from the recent studies. That was very nice. So it was not just the history, but you developed that entire session coming till the recent. And I'm very sure, as Springer has already given us the free download, we all will get an opportunity to read your book.
Yeah, sure. Yeah.
So,
In
Praveen,
fact,
sir,
I must say that there is one section I intentionally included there as future of enzymology, which probably you should look at, a little more details as to where we stand today. Yeah.
Thank you, sir.
Okay. Yeah. Okay. Yeah.
Praveen, sir?
Yeah.
Yeah. Praveen, sir.
Yeah.
Yes, please propose the vote of thanks.
Yes, ma'am. Yeah, sure. It's a profound, task given to me to propose a vote of thanks. So on behalf of Association of Teachers in Biological Sciences, ATBS, and Department of Biological Sciences, Ram Niranjan Jhunjhunwala College, I extend a warm our gratitude and thanks to Punekar sir for giving a very lucid and simple talk covering all the aspects of the enzyme catalysis in addition to giving a touch to the modern scope or future scope of enzymology. So thank you very much, sir. I would also like to take this opportunity to thank our teacher, Dr. Karmakar sir, who has been attending all the lectures right from the start. So thank you very much, sir. I can see on my screen a number of teachers like Nath sir. Nath sir also has been consistent in attending a number of lectures. Malik sir, Balaji sir. Okay, so thank you very much, sirs, for attending this session. So on behalf of ATBS and Junjunwala College, I thank all the ATBS members, teacher colleagues of this college, the RJ college, as well as of various other colleges, students of this college as well as other colleges for attending and listening to this lecture series. Not last but not the least, I'd like to thank Mr. Devi Prasad Shetty for providing the technical assistance throughout, not only for this particular lecture, but also throughout the lecture series. So I thank one and all. Thank you, sir. Madam.
Thank you, sir.
Yeah, thank you so much. See you tomorrow in the evening.
Yeah. Bye.
Yes. Thanks.
Thank you, everybody.
DP sir, please end the session.